Hello, everybody. We're going to wait just a few more moments to see if anybody else plans to join the webinar today. So hang in for just a couple more minutes. I believe attendance has slowed down, so we should be good to start. Thank you, everybody, for coming to our webinar today. We're going to be reviewing how to read a genetic test report. All of our webinars are recorded and available on our YouTube playlist. The list of recordings is on, like I said, YouTube and can be accessed using the QR code on the slide. For those of you who provided questions beforehand as part of your registration, we thank you for that. Those have been tried to be incorporated into today's webinar presentation. We hope to answer majority of your questions throughout as well. We will have time at the end to take some of the questions. If you have questions during this webinar, please use the Q&A box located at the bottom of the screen. We are reviewing example reports in general today, so these are not specific to any one person's genetic testing, and we ask that you do not put your private genetic information into the chat as this is recorded. So this is our webinar team today. We have myself, I'm Alexis, one of the genetic counselors with ClinGen and GenomeConnect. We are joined by two of our other genetic counselors, Caitlin and Jules, and one of our genetic counseling assistants, Gabrielle. During the webinar today, we will start with an overview of different genetic concepts to assist in our main discussion, which will be reviewing two different genetic test reports, a microarray report and a sequence report. So individuals are made up of millions of cells, and those cells contain chromosomes. Chromosomes are structures that hold all of our DNA. Our DNA make up our genes, and each gene is an individual instruction for our body. Typically, people have 46 chromosomes total. Our chromosomes are numbered from biggest to smallest for the first 22 pairs. The first 22 pairs are called autosomes. Typically, a person gets one copy of each chromosome from each biological parent. The last set of chromosomes are our sex chromosomes. Typically, people have either two X chromosomes or one X and one Y. Wrapped up tightly in those chromosomes is our DNA. Our DNA provides instructions to make proteins. Each instruction is a gene. The proteins play important roles throughout the body, and each protein has a different role in the body. Our DNA is made up of letters called nucleotides. There are 4 different nucleotides. We have A, T, G, and C. Our body reads these nucleotides to make proteins. The nucleotides are read in sets of 3. So we can see on the screen we have here grouped in 3 across. So a set of 3 nucleotides codes for 1 protein building block within the whole protein chain. So here we have the 3 nucleotides and then that one part of the protein chain itself. These building blocks are called amino acids. The example on the slide, the DNA sequence starts with A, T, G, which is that first set of nucleotides that codes for one of the protein building blocks of, like I said, this entire protein chain. We all have changes in our DNA, which we call variants. These are part of what make us who we are. Some variants can affect our health, development, or risk for future health concerns. And a person can have different types of genetic variants. We can have variants at the chromosome or the gene level. Different types of genetic tests will detect different types of genetic variants. A karyotype is the first test we'll talk about. This looks at our chromosomes in big picture, while a microarray zooms in more detail of the chromosomes. And then a sequencing test zooms in even further for the most detail. You can think of these different genetic tests as a book set. A karyotype is the test that looks to see if you have every book in the set. A microarray looks at each book's chapter to see if all the chapters are there. And sequencing tests look at the sentences on the pages to see if there are any changes to the sentences and letters. There are different types of sequence genetic test reports and tests as well, the main difference being how many genes the test is looking at. Whenever a genetic variant is found, a genetic testing laboratory tries to classify its effect on health. Variants are classified into different buckets. We have benign, likely benign, uncertain or unclear, and pathogenic, likely pathogenic. Benign, likely benign means this genetic variant is not thought to impact gene function. Therefore, it's not expected to cause disease. Uncertain or unclear means there's not enough information about this variant to know its impact on health. We don't know if this can cause disease or if it's a normal genetic change anybody can have. Additional information over time may be able to help better understand these uncertain variants. Pathogenic, likely pathogenic means this genetic variant is known to impact gene function and contribute to disease. These types of variants may explain a person's symptoms, increase a person's chance to develop a certain condition, or increase the chance of having a child with a certain condition. When a genetic testing laboratory classifies a variant, they use many pieces of information to classify the variant into one of these buckets. For example, has this variant been seen in the general population? Was this variant inherited from a parent? What's the impact on the protein for this variant? And many more questions. Depending on the information available, variants will then be classified into one of these buckets. As more information becomes available, a variant's classification may change. Any variant in any bucket has the potential to change. So for example, a pathogenic variant might be downgraded to likely pathogenic, or an uncertain result might be upgraded to pathogenic, or possibly downgraded to benign, likely benign. In fact, we hope to reclassify uncertain variants into a more clear category. Not every variant will be reclassified over time. There is not a timeline for reclassification. Some uncertain results may never be reclassified. Some might be reclassified in a month. Others may still be uncertain after years. So now that we have reviewed some genetic concepts, let's start to review some example genetic test reports. And first, we're going to start with that example microarray report. So this specific test looks at an individual's chromosomes to detect full missing or full extra chromosomes, called monosomy and trisomy respectively. A microarray also looks within the chromosomes for big or small missing or extra pieces of a chromosome called deletions or duplications. Together, these chromosome variants are called copy number variants or CNVs. So this is an example microarray report that looks similar to many other microarray reports, and we will review what each part of this report means. So starting with the result itself, if there is a chromosome variant identified, the result may say positive or uncertain. If there is no chromosome variant identified, it will likely say negative. For this example report, it says positive for a heterozygous 1.65 megabase deletion at 16p13.11. This is a lot of information, so we will break down each part. Each chromosome has two arms, a short arm called the P arm on the top of the chromosome, and a long arm called the Q arm on the bottom of the chromosome. And each chromosome has a specific banding pattern, which are the stripes you see throughout the chromosome. This report is stating that there is a copy number variant on one of the 16th chromosomes. Specifically, there's a deletion on that P arm or the short arm on top, located at the band 13.11, which we have circled in that black circle. The report will also note the classification of that specific copy number variant. For this example report, it states that this copy number variant is pathogenic, or the variant is known to be associated with a condition. Another way you might see a copy number variant written is shown in this circle. So ARR means this is a microarray. GRCH37 is stating the specific genomic database or the genomic build that was used for this test specifically. You may see the genomic build as 38 or possibly even 36 if it was done a few years ago. Then it will state the chromosome and the banding pattern. So for this example, 16p13.11. The numbers in the parentheses after are giving the specific genomic coordinates that the copy number is affecting. So the first number is the starting point, and the last number is the ending point. So from that start to the end is where that piece of information is missing or deleted. Next, you will see an X and a number. This tells you how many times this specific copy number was seen. Typically, a person would have two of these regions, one on each chromosome. But in this example, the one piece is deleted, so we only see this region one time, which is why it is X1. If it's typical for there to be two regions, but someone has a duplication, it would say ×3. Keep in mind that ×1 and ×3 are not the only numbers that you may see. Sometimes you may see ×0, ×2, possibly ×4. Another thing you may see at the end of this section is the word MAT or PAT. That is only included if parental samples were sent for testing as well. So if it says MAT or PAT, that tells you that this copy number variant was inherited from a biological parent. Reports will also have a results and interpretation section where they provide additional information about what type of copy number variant was detected, what genes are included in that chromosomal region, and what conditions might be caused by that copy number variant. This specific example states this deletion is consistent with a diagnosis of 16p13.11 microdeletion syndrome. If parental samples were provided for testing as well, the report will note if this specific copy number variant was inherited from a parent. In this example, it states that it is de novo, meaning the copy number variant was not inherited from either biological parent, but was a brand new change in this individual. When a variant is de novo or brand new, that means neither biological parent has that variant. So in this picture, we don't see that star suggesting that variant in either parent, but the child does have that star. So next we will review how to read a sequencing report. A sequencing test looks at an individual's genetic material in more detail. It's looking at the gene level for any changes in the letter of the DNA, what we sometimes can call spelling in the genes. These types of genetic changes are called sequence variants. Looking at the example on the slide, there is a DNA sequence, perhaps a portion of one gene. The top letters are the typical spelling for this gene. In the patient sequence, you can see a sequence variant where one of these letters changes to another letter. So in this example, where we see a T nucleotide typically, we see a C nucleotide in this patient sequence. So this is an example report for a sequence report that's meant to look similar to most other sequence reports. And same thing as that microarray, you will review what each part of this report means. So these reports will mention the gene that was identified to have a sequence variant. In this example, the gene is USH2A for both variants in this example report. We can also see at the top of the report what test was ordered. So in this instance, it was a gene panel. These gene panels are usually specific to what type of symptoms or the reason for testing. For this individual, being congenital deafness/hearing loss, as well as vision loss. Sequence reports will also note if there is a disease or a genetic condition associated with that gene. In this example, the USH2A gene is associated with a vision loss and hearing loss condition called Usher syndrome. Not every gene will have a condition associated with it. Some genes may not have a condition and might be called genes of uncertain significance or candidate genes, meaning we don't understand the gene's relationship to disease, but based on some information, it could be tied to health, development, or disease. While some genes might not yet be tied to a condition, other genes might actually have multiple conditions associated with it. The variant information of the sequence report tells you exactly what genetic change or sequence variant was identified through testing. You will typically see a C dot and a P dot on the reports. The C dot is the DNA information, and the P dot is the protein information. So in this example, we're looking at the first variant. The C dot or sequence variant is 2276G to T. So this means the number 2276 is the nucleotide position, which means at the 2276th letter of the USH2A gene, that letter or nucleotide is typically a G. That G is called the reference nucleotide, but in this individual, that G is actually a T at that position. That T is known as the variant nucleotide. The P dot or protein description tells us what amino acid is typically in that position of the protein and how the protein is changed. So missense variant is a DNA change that is often caused by a single letter change. This is the example that we just showed on the previous slide. So the single letter change changes a single amino acid in the protein. So in this example, the G was changed to a T in the DNA sequence, which changes the amino acid that is typically cysteine to phenylalanine. And that change may impact how the protein functions in the body. So to summarize, this missense variant means there are a change to a DNA sequence that changes the amino acid in that protein. And this change can affect the protein's function in the body. In the second example, the C dot or sequence variant is 99_100 INST. The numbers 99_100 is the nucleotide position, meaning in between the 99th and 100th nucleotide, of the USH2 gene, there's an extra letter. So that's what the INS stands for, that insertion. And that letter that's inserted is T in this example. Usually there's no T in that position, so we have that extra nucleotide added in. The P dot, or protein description, tells us what amino acid is typically in that position of the protein and how the protein is changed. So same as that previous variant is what the P dot is telling us as well. Although this is a different type of sequence variant, so therefore the impact on the protein is slightly different. So when we were reviewing the microarray report, we talked about large deletions and duplications in the chromosomes. Those deletions and duplications are different from these types of deletions and duplications. These sequence variants are usually smaller deletions and duplications within the DNA sequence. Adding and removing letters in the DNA sequence can result in two different types of variants. The first is what we call a frame shift variant, which is the example in our report. And then the second example on our report, like I said, is this frame shift variant. The other type is called an in-frame variant, which we will review in a moment. So we talked earlier that the body reads DNA sequence in sets of 3. Insertions, duplications, or deletions of the DNA. So any change to the DNA sequence that's not in a set of 3 will shift how our body reads the remaining letters and can change the remaining amino acids, eventually resulting in an early stop to the protein. Since it's changing the reading frame, it's called a frame shift variant. You can identify frame shift variants because they typically have FS in the description of the protein change. So on the slide at the bottom, this is the specific protein change from our example report. So we can see that FS at the end suggesting this is a frame shift variant. So in this example that we're reviewing, it's an insertion of one nucleotide, which again is noted by the INS-T in the C dot. This is not a set of 3, so it changed the reading frame of the DNA. Therefore, changing the protein, which is noted in that P dot. The protein changes and is shortened, so we see that FS at the end, again suggesting frame shift variant. There are different types of sequence variants, and different sequence variants may have different impacts on the protein. So this slide just gives a brief description of different types of variants. We're going to review these variants in a little bit more detail in upcoming slides, but we also have videos on our YouTube channel that reviews these types of sequence variants in more detail, and that can be found using the QR code on the slide. So this specific example variant is called a nonsense variant. In this example, at this position, the typical nucleotide is a G. In this individual, they have a T nucleotide at the position instead of a G. In addition to just coding for amino acids in the protein building blocks, some groups of 3 nucleotides tell the body when the protein should stop. These are called stop codons. This specific change to the DNA sequence creates a stop codon instead of a typical arginine, therefore stopping the growth of the protein. Now this individual has a shorter protein chain, which impacts the protein's function in the body. So to summarize, nonsense variants means there is a single letter change to the DNA sequence, and that single letter change changes the protein to stop early, therefore affecting the body's protein function. Synonymous variants, also known as silent variants, are just that. The change to the DNA sequence does not cause a change to the amino acid. Therefore, it does not cause a change to the protein. So in this example, The sequence nucleotide C was changed to a T, but both TCC and TCT code for the same amino acid, serine. Since they both code for the same amino acid, it doesn't change that amino acid. Therefore, it doesn't change the protein, and the protein continues to function as expected. These types of variants are not expected to be associated with disease and are generally benign variants that anybody can have. So to summarize, a synonymous or silent variant means there is a change to the DNA sequence, but this change does not impact protein, and these variants are benign and do not cause disease. We did just review the frameshift variant in our example report, but just to reiterate, that frameshift variant is an insertion, duplication, or deletion of the DNA, not in a set of 3. Therefore, shifting how that body reads the remaining letters and can change the remaining amino acids, eventually resulting in that early stop to the protein. So in our example report, we had an insertion of 1 nucleotide. In this example, we have an insertion of 13. So 13 of these were duplicated, so we're seeing that sequence twice. And then this is our second type of variant that we can get when it's not shifted in that 3. So that first change we talked about was the frameshift variant. The second is that in-frame variant. So insertions, duplications, or deletions of the DNA in sets of 3 does not shift how our body reads the remaining letters. Since it is not changing the reading frame itself, it's called an in-frame variant. The example on the slide is a deletion of 3 nucleotides. So it did not change the reading frame, but since there was a deletion of one of the amino acids, it still did change the protein, which is noted in the P dot. So the protein changed and an amino acid was deleted, which is denoted by the del at the end of the P dot. With both frameshift variants and in-frame variants, we can have deletions and duplications. We can also have an insertion of nucleotides, as well as indels, which is in a section of a sequence, is deleted and replaced by a new sequence. These different types of variants can result in either frame shifts or in-frame variants. The difference between in-frame and frame shift variants is solely if the DNA change is in sets of 3 or not. Our DNA has both exons and introns. Exons are the coding portions of the gene, and introns are the non-coding portions of the gene. The introns get cut out of the gene in order for the gene to produce the protein. So splicing is how those introns are cut out from the exons. There are DNA sequences that can help recognize the boundary between exons and introns for splicing to happen. This can be compared to drawings in school with the dotted lines showing you to cut here. A genetic variant in the DNA sequence that affects splicing is called a splice site variant. This type of variant disrupts the RNA splicing and can cause loss of exons or introns that don't get cut out, so they are included, which can all impact the protein. In this example, the body typically recognizes a splice site between G and G, but here the second G is changed to a T, so the body is not able to read that splice site anymore and is therefore missed. So on a report, this would show as a plus or a minus with a number after. So for example, +1, -1, +2, -2, etc. on a genetic test report in the cDNA. These types of variants have an impact on the protein by either shortening or lengthening the protein, which both may impact the protein's function. So now that we've reviewed some of those genetic variants that we can see, we'll go back to the report. So the next section to talk about is the inheritance pattern. The inheritance pattern helps us understand how many copies of the specific gene needs to be not working in order for somebody to either have or be at risk for the specific genetic condition. Inheritance patterns don't refer to the specific patient and don't tell us if this variant was inherited from a parent. So in this example, USH2A gene is associated with an autosomal recessive condition. That's why it says inheritance pattern autosomal recessive. With autosomal recessive conditions, this means typically both copies of the gene have to have a pathogenic or likely pathogenic variant in both of the copies to cause symptoms or have features of that condition. When people have just one variant in an autosomal recessive condition, we call that person a carrier, as they are typically not at risk for the condition itself, but just a silent carrier. Whereas for autosomal dominant conditions, that means out of the two copies of the genes we have, a pathogenic or likely pathogenic variant in only one of those gene copies is enough for a person to be at risk or have symptoms or features of the condition. X-linked is another common type of inheritance pattern that you can see on reports. For X-linked conditions, that means the gene that causes the condition is located on the X sex chromosome. Symptoms in people who have variants on their X chromosome can differ between individuals who have two X chromosomes compared to those who have one X chromosome and one Y chromosome. This was not an extensive list of inheritance patterns. We are discussing the more common ones. If you want to learn about the less common inheritance patterns, you can always do so on our YouTube video series. So the next section is zygosity. Zygosity tells us if the sequence variant was found on one or both copies of the gene. So this does tell us more about the specific patient. This example report tells us the patient is heterozygous, which is used to describe when that one variant was found in the one copy of the gene. So in this example, there are two different variants in the same gene. Therefore, this individual is heterozygous for each variant in the USH2A gene. As I said in our example report, this individual would actually be considered a compound heterozygous, which means there were two different variants in the same gene, again, as the example in our report. Some of the other terms you might hear include single heterozygous, homozygous, or hemizygous. Some reports will say single heterozygous, where others will just say heterozygous. These mean the same thing, that the variant was only found in one copy of the gene. Homozygous means the same variant was found in both copies of the same gene. And hemizygous is used when one variant was found in a gene, but that person only has one copy of the gene. This is usually only used for conditions on the X chromosome. So for somebody who has a gene change on their only X chromosome, they would be considered hemizygous. Moving on to inherited from. As mentioned before, inherited from is different from inheritance pattern. Inherited from tells us if this sequence variant came from one or both parents, or if the sequence variant was a brand new change in this individual. So in this example, the first sequence variant is maternally inherited. And the second sequence variant is paternally inherited. So there is a variant inherited from each biological parent. Maternal means the variant was inherited from the biological mother, and paternal means the variant was inherited from the biological father. So in this example, the biological parents are not affected by the condition, but are carriers for Usher syndrome. And our last section is variant classification. So on sequence reports, they will provide the variant classification. This tells you how the laboratory thinks this variant may impact a person's health, and if the variant may cause the associated genetic condition. So in this first example variant, the classification is likely pathogenic. And the second variant example is classified as pathogenic, meaning both of these variants are expected to be associated with Usher syndrome. Therefore, that individual is at risk for vision loss and hearing loss. So both of the reports that we reviewed today had either pathogenic or likely pathogenic variants identified. And we wanted to take some time now to talk about those variants of uncertain significance. So to reiterate, reclassification of variants, which we briefly discussed earlier in the webinar. So as mentioned, genetic labs will try to determine if a variant impacts health or disease risk. The labs explore if the effects of the variant have been studied in a lab, if the variant was inherited from a parent, if similar variants have been found and reported in other individuals, if people in the general population are found to have this variant. They ask questions such as, has this variant been seen in people with symptoms? And what's the impact on the protein with this variant? So once the lab looks into all of these questions, they will then classify the variant as pathogenic, likely pathogenic, unknown or uncertain, likely benign or benign. Variants of uncertain or unknown significance do not have enough evidence at the time to determine whether these variants cause health differences or are variants anybody can have and not related to health differences. The goal with uncertain variants is to learn more about them over time, so we can better understand if they have a relationship with disease or are a variant anybody can have, and again, not associated with disease. As we learn more about a variant, the lab may be able to downgrade an uncertain variant to likely benign or benign, but they may also learn more information that allows the lab to upgrade it to likely pathogenic or pathogenic. Just to reiterate, any variant in any bucket has the potential to move. So a pathogenic variant might be downgraded to likely pathogenic, or an uncertain result might be upgraded to pathogenic, but also could be downgraded to likely benign. In fact, we hope to reclassify these uncertain variants into a more clear category. Not every variant will be reclassified over time. If you want to know if there has been any changes to a genetic test report for yourself or for a family member, we would encourage you to reach out to the ordering provider to learn if there have been any reported updates. Or you could reach out to a local genetics provider. And sometimes you can reach out to the laboratory itself that completed the testing for more information as well. So we would like to thank you all for joining today's webinar. We hope this has been helpful. Additional information about all of these topics can be found on our YouTube channel videos. As a reminder, this webinar was recorded and will be available after today as well. At this time, I'm going to open up our Q&A where Jules, Caitlin, and I will all take some questions.
Thanks, Alexis, for that presentation. And I think you did a lovely job answering some of the questions that we got before today's webinar. So thanks if you shared those with us. Feel free if you have questions now to put those in the question and answer. While we wait for some questions to come in, I'll start with some other questions that I think came through via the registration link or that we often hear. So one question was around negative results and what they might mean for a family. So I know you spent a lot of time on variant classifications, but what do negative results mean?
So negative results always have to be kind of interpreted in context of a few things. So one, the individual and family's history and any of their clinical symptoms they have, and also in context to the panel or the specific test that they have had. So one case that's an easy example that I'll start off with, if we know that there is a specific change in the family, say that there's a family member with cancer and they had genetic testing and they found a hereditary cancer variant, we can go on and test family members for that specific variant to see if they have inherited that change or if they don't, and that would provide information in terms of their own screening and care recommendations. Other times, if we're testing an individual that kind of has a number of symptoms, a number of clinical presentations, sometimes a doctor or a genetics provider will start with a smaller test. If that comes back as negative, they may move on to a larger test, meaning we may start with a panel, as Alexis said, that only will look at a few genes that maybe have that overlap with the presentation, or maybe they would move on to looking at all of the exons, which are all the coding portions of our genes. And then currently the largest test that we have available right now is Genome, which is looking at all of the coding and non-coding portions of the gene. So a negative result, it has can be a little bit hard to just put overall in a bucket of what it means. I really need to put in some of those other pieces of the family history, the clinical presentation, and what are we looking to gain with testing?
Thanks for that, Caitlin. We had another question come in the Q&A. So Alexis, I might tee this one to you if sounds okay. But they're asking, what is germline mosaicism?
That is a great question, and we actually have some slides that might be able to help with this, so bear with me while I pull those slides up. Okay, so I will start by discussing mosaicism in general and then move to germline mosaicism. So mosaicism means that some of a person's cells has that specific genetic change, but some of the cells don't have that genetic change. So in this picture, we can see some blue cells that don't have the genetic change, but then we can see some yellow cells that do. So mosaicism in general might mean that person is mosaic throughout their entire body. Versus germline mosaicism is when that person just has that genetic change in their germline, aka their sperm or their eggs. Not all of the sperm or eggs would have that genetic change, but some of them can. So in this picture, we can see that the individual, so the black circles were circling the germline area. Some of the germline cells or eggs in this case have blue and they have no genetic change, but then there are a few that are yellow and have that genetic change. So when a person is germline mosaic, they still could have an increased chance of having multiple children with that genetic change in their entire body then.
Thanks for that answer, Alexis. I think a follow-up that we didn't hear in the Q&A today, but that I often hear is there testing available to figure out if I'm a germline mosaic? So I know you wear some clinical hats and probably get that question in other venues. So I'm wondering if you could speak to that.
Yeah, it's very challenging to do germline specific testing for individuals because it requires us to have egg or sperm to do genetic testing on. There are specific genetic changes where if it's not found in either biological parent, might have that slightly higher chance of being mosaic in just the sperm or egg. So usually that's why we say if a genetic change was de novo or brand new in a child, there always could be that slight chance of having another child with the same condition because we just can't rule out that germline mosaicism.
Thanks, Alexis, for the follow-on. We also have a question in the chat of whether those with recessive genes ever display some, maybe more mild symptoms. So I think, and got this question from registration as well, really around, does everyone with the same condition have the same symptoms? If you have the same variant, do you even have the same symptoms? Caitlin, you want to take this one? I'm happy to, if not.
Yes, of course. So great question, and a little bit of it depends is the answer, which I know is kind of unclear, but for some of the recessive conditions, there are some specific variant changes in certain genes in which the individual who is a carrier may exhibit, as you alluded to in the question, mild symptoms, may present with a little bit different clinical presentation than if someone has the two non-working copies of the gene. The majority of the time, individuals who are carriers for a condition will not have symptoms, but there are a few conditions in which we do see carriers, they may have some milder presentation, some impact on their health. In terms of if I have the same variant as, my neighbor, will I present exactly the same? And again, it depends, and that is most likely going to be unlikely. The truth is we have 25,000 genes, and so it is hard to know how all of them are working together and how all of them are combining in terms of our proteins and how all of them are functioning. So we do say that for a number of conditions, there is kind of a variable expressivity, meaning multiple symptoms. Not everyone has all 10 symptoms. Some people may have 2, some people may have 5. And this can vary not just between you and your neighbor, but can also vary within the family. And so that's sometimes can make things a little bit hard to say, oh, I have it. It's going to be exactly the same for this person and this person. Each person can have a little bit different a clinical impact, a clinical presentation.
Thanks for that, Caitlin. And certainly there are some conditions where we might say, well, certain types of genetic changes like missense might be more mild typically, but always all those caveats that Caitlin talked about of depends on the condition, still is going to be variability, all those things. Another question in the chat. You mentioned what plus and minus means, but what does the greater than or the alligator or carrot, depending on what you learned in grade school, mean? The T was replaced with a C, for example, and the T greater than C. So Alexis, could you just refresh us there?
Yes, so if it says T greater than C, that means it's originally a T and was replaced by a C. So that greater than or the caret sign is telling us what it is replaced with. The number or the letter before the sign telling us what it is originally supposed to be, the letter after suggesting what it has been replaced with.
Thanks for that, Alexis. And I think our last question, and then we'll close us out for the day. Caitlin, can I give this one to you? Can our genes change over time? And I know it's a complicated answer as well. So I think I've given you all of the sort of kind of it depends question. So here's another one.
Yeah, so can our genes change over time? So a lot of the testing that we focused on today is what I call germline testing, which would be the genetic information we were born with. And that usually does not change over time. This is the DNA kind of within each cell of your body. What can change is certain what we would call somatic changes, changes in some of the cells over the course of our life. So not necessarily the cells that the DNA code that we were born with. I'm going to go back to my example. A lot of times when we see cancerous cells are related to that there are changes in the genetic code that happen over the course of our life. And so someone recently told me this really great example, which I'm going to share with you. It's kind of when we're born with all our genetic information, and our cells have to copy our information over the course of our life. So every time we make a new cell, we are copying all of our genetic information, and we are doing this each time we're making a new cell. So making copies of copies of copies. So kind of if you take a screenshot of an image on your phone and you send it to someone, and then they take a screenshot, and then they take a screenshot and another screenshot, and we keep sending it, we know that the image is going to get a little bit more unclear, a little bit fuzzy, a little bit smaller. So we can see that some of the changes in our genetic code that can happen over the course of time, and that's due to age, environmental factors, lifestyle factors. So those things can change, but those changes are not necessarily passed down from one generation to the next. Those are changes in a few cells of our body.
Thanks for that, Caitlin. And I think we'll leave it there for today. There are a couple of other questions in the chat that I will hope maybe we can stay on for another minute and type a few answers, but I think we can let the attendees go. Just thank you so much, Alexis, for the presentation today. And thank you, Caitlin, for helping to answer those questions that were asked of the team today.
Thank you, everyone. And as we mentioned, this webinar will be on our YouTube channel in the next week or so. So it will be available there as well.
Thanks so much. Have a good rest of your afternoon, everyone, and happy DNA Day.
Thank you.